Have you ever wondered about the lives of people who lived thousands of years ago? What did they look like? What did they eat? Where did they come from? Amazingly, we can find answers to these questions by studying ancient DNA. Ancient DNA, or aDNA for short, is the DNA extracted from ancient remains. It's like a genetic time capsule holding clues about the past. This DNA can come from humans, animals, or even plants. By analyzing aDNA, scientists can learn incredible things about our ancestors and the world they lived in. Think of aDNA as a history book written in a secret code. This code is made up of DNA sequences, the building blocks of life. By deciphering this code, scientists can unlock secrets hidden for millennia. Through aDNA, we can trace human migration patterns, understand the evolution of diseases, and even reconstruct the appearances of our ancestors. It's a powerful tool that allows us to peer into the past and unravel the mysteries of our origins. Ancient DNA is a precious and fragile treasure, often hidden in unexpected places. This genetic material, known as aDNA, offers a unique window into the past, allowing us to uncover secrets about ancient organisms, their environments, and even their interactions. But where exactly can we find these remnants of the past? The answer lies in materials that preserve genetic information over long periods. Skeletal remains, like bones and teeth, are the most common sources of aDNA. The hard structure of bones helps protect DNA from degradation, offering a glimpse into the genetic makeup of individuals from long ago. Teeth are particularly valuable as they contain enamel, which provides even better DNA preservation. This makes them a goldmine for geneticists looking to piece together the biological history of ancient populations. But aDNA is not limited to bones and teeth. Scientists have also extracted aDNA from mummified tissues, preserved in arid environments, or through intentional mummification practices. These tissues, such as skin, hair, and organs, can provide even more detailed genetic information. Mummified remains are particularly fascinating because they often preserve soft tissues that are rarely found in other contexts. This allows researchers to study not just the genetic material but also the physical characteristics and health conditions of ancient individuals. Surprisingly, aDNA can also be found in sediments, even without any skeletal remains. Over time, DNA from organisms living in an area can leach into the surrounding soil and be preserved. This environmental DNA allows scientists to reconstruct past ecosystems and identify the plants and animals that once inhabited a region. This method of studying ancient environments is particularly useful in areas where traditional fossils are scarce. By analyzing soil samples, researchers can gain insights into the biodiversity and ecological dynamics of ancient landscapes. Even ice cores, drilled from glaciers and ice sheets, can contain aDNA. As ice forms, it traps air bubbles and particles, including DNA from organisms that lived in the past. These icy archives provide a snapshot of ancient environments and the organisms that thrived in them. The study of aDNA from ice cores is particularly important for understanding climate change. By examining the genetic material trapped in ice, scientists can track how populations of plants and animals have responded to past climate shifts, offering clues about how current and future changes might impact biodiversity. In addition to these sources, aDNA has also been found in other unexpected places, such as ancient dental plaque and coprolites or fossilized feces. These materials can provide unique insights into the diets, health, and lifestyles of ancient peoples and animals. The field of ancient DNA research is continually evolving, with new techniques and technologies allowing scientists to extract and analyze genetic material from increasingly older and more degraded samples. This ongoing research is helping to fill in the gaps in our understanding of the past, providing a more complete picture of the history of life on Earth. As we continue to explore and discover new sources of ancient DNA, we are constantly reminded of the incredible resilience of genetic material and its ability to survive through the ages. Each new discovery brings us one step closer to unraveling the mysteries of our ancient world. From the bones and teeth of long-dead individuals to the sediments and ice that have captured the essence of ancient ecosystems, the quest to find and study ancient DNA is a journey through time itself. It is a testament to the enduring power of science to unlock the secrets of our past and illuminate the path to our future. The study of ancient DNA is not just about understanding where we come from, but also about learning how life has adapted and evolved over millennia. It is a field that bridges the past and the present, offering insights that are crucial for our future. As we delve deeper into the genetic archives of our planet, we uncover stories that have been waiting millions of years to be told. These stories are not just about the organisms that once lived, but also about the environments they inhabited and the changes they endured. In the end, the search for ancient DNA is a journey of discovery, one that connects us to the vast tapestry of life that has unfolded on Earth. It is a reminder of our shared heritage and the enduring legacy of the natural world. The study of ancient DNA is a relatively new field, but its impact on our understanding of the past has been revolutionary. By delving into the genetic material preserved in ancient remains, scientists have been able to unlock secrets that were once thought lost to history. This field, known as ancient DNA or ADNA research, has provided us with unprecedented insights into the lives of our ancestors, the evolution of species, and the migration patterns of ancient populations. The journey began in the 1980s, with scientists taking the first tentative steps towards recovering DNA from ancient samples. During this pioneering era, researchers faced numerous challenges, including contamination and degradation of the genetic material. Despite these obstacles, the determination and ingenuity of these early scientists laid the groundwork for future breakthroughs. Early attempts focused on extracting DNA from mummified remains, such as Egyptian mummies. These ancient relics, preserved through the meticulous process of mummification, offered a unique opportunity to study DNA that had been shielded from the ravages of time. The successful extraction of DNA from these mummies was a monumental achievement, sparking excitement and curiosity within the scientific community. These initial studies were met with excitement but also skepticism, as doubts lingered about the authenticity and reliability of the results. Critics questioned whether the DNA recovered was truly ancient, or if it had been contaminated by modern sources. Rigorous testing and validation were required to address these concerns, leading to the development of more stringent protocols and techniques. A major breakthrough came in the late 1980s with the development of a technique called polymerase chain reaction, or PCR. This revolutionary method allowed scientists to amplify tiny amounts of DNA into millions of copies, making it possible to study genetic material that was previously too scarce to analyze. PCR opened new doors for ADNA research, enabling the examination of a wider range of ancient samples. PCR acts like a molecular copying machine, allowing scientists to amplify tiny amounts of DNA into millions of copies. This process not only increased the quantity of DNA available for study, but also improved the accuracy and reliability of the results. With PCR, researchers could delve deeper into the genetic history of ancient organisms, uncovering details that were previously hidden. This revolutionized the field of ADNA, making it possible to study even highly degraded samples. Ancient bones, teeth, and other remains that had been exposed to harsh environmental conditions could now be analyzed with greater precision. The ability to work with degraded DNA expanded the scope of ADNA research, allowing scientists to explore a broader range of historical and prehistorical contexts. The 1990s saw a surge in ADNA research, with scientists extracting DNA from a wider range of sources, including Neanderthal bones. These studies provided the first glimpses into the genetic makeup of our extinct human relatives, challenging our understanding of human evolution. The discovery of Neanderthal DNA in modern humans revealed a complex history of interbreeding and migration, reshaping our view of the past. These studies provided the first glimpses into the genetic makeup of our extinct human relatives, challenging our understanding of human evolution. The genetic data obtained from Neanderthal remains offered new perspectives on their biology, behavior, and interactions with early modern humans. This research not only deepened our knowledge of Neanderthals but also highlighted the interconnectedness of all human populations. As technology advanced, so too did the field of ADNA. The development of more sophisticated tools and techniques allowed scientists to extract and analyze DNA with greater efficiency and accuracy. Innovations in laboratory equipment and computational methods transformed ADNA research, making it possible to tackle more complex questions in larger data sets. Next-generation sequencing technologies, developed in the 21st century, enabled scientists to sequence millions of DNA fragments simultaneously. This high-throughput approach dramatically increased the speed and affordability of ADNA analysis, ushering in a new era of discovery. Researchers could now generate vast amounts of genetic data in a fraction of the time it previously took, opening up new possibilities for studying ancient genomes. This high-throughput sequencing dramatically increased the speed and affordability of aDNA analysis, ushering in a new era of discovery. The ability to rapidly sequence and analyze large volumes of DNA data has led to groundbreaking findings in fields ranging from archaeology to evolutionary biology. As we continue to refine our techniques and expand our knowledge, the study of ancient DNA promises to reveal even more about the intricate tapestry of life on Earth. The early years of ancient DNA, or aDNA, research were marked by groundbreaking discoveries that reshaped our understanding of the past. These early efforts laid the foundation for a new field of study, one that would bridge the gap between the present and the distant past, offering unprecedented insights into the lives of long-extinct species and ancient human populations. One of the first major breakthroughs came with the study of the quagga, an extinct zebra subspecies. The quagga, which once roamed the plains of South Africa, had been extinct since the late 19th century. Its unique appearance, with stripes only on the front half of its body, made it a subject of fascination for scientists and the public alike. In 1984, scientists successfully extracted DNA from a 140-year-old quagga museum specimen. This remarkable achievement was a pivotal moment in the field of aDNA research. It demonstrated that genetic material could be preserved over long periods, even in specimens that had been stored in less than ideal conditions. This was the first time DNA had been retrieved from an extinct creature, demonstrating the potential of aDNA to shed light on lost worlds. The success with the quagga opened the door to a new era of research, where scientists could explore the genetic makeup of other extinct species, providing a window into ecosystems that no longer exist. Another significant discovery came with the analysis of aDNA from the original Neanderthal specimen, discovered in Germany in 1856. The Neanderthals, our closest extinct relatives, had long been a subject of intense study and debate. The ability to analyze their DNA provided a new dimension to our understanding of their biology and their relationship to modern humans. By comparing Neanderthal DNA to modern human DNA, scientists gained crucial insights into the relationship between our two lineages. This comparison revealed that Neanderthals and modern humans shared a common ancestor, and that there had been interbreeding between the two groups. These findings challenged previous notions of human evolution and highlighted the complex web of interactions that shaped our species. aDNA also revolutionized our understanding of ancient human migrations. The movement of ancient populations had always been a topic of great interest, but traditional archaeological methods could only provide part of the picture. With aDNA, scientists could trace the genetic footprints of these migrations, offering a more detailed and accurate reconstruction of how humans spread across the globe. By analyzing aDNA from archaeological sites across the globe, scientists were able to trace the movement of populations and reconstruct the peopling of the world. This genetic evidence complemented and sometimes contradicted archaeological findings, leading to new theories and a deeper understanding of our shared history. One such study revealed that early farmers from Anatolia, modern-day Turkey, migrated across Europe around 7,000 years ago, spreading their agricultural practices and genes. This migration had a profound impact on the genetic landscape of Europe, as these early farmers intermingled with local hunter-gatherer populations. This contradicted the previous belief that agriculture spread through cultural diffusion, highlighting the power of aDNA to rewrite history. The ability to extract and analyze ancient DNA has transformed our understanding of the past, providing a powerful tool to uncover the hidden stories of our ancestors and the world they inhabited.